TAS is a spectroscopic measurement that gives you information of photoexcited species because it probes photoexcited species. So it measures the absorbance of these excited species. So also another good thing about this technique is like it measures the evolution of these photoexcited species with time. And as I said, is related to the absorbance of these species. So it's going to follow the Lambert-B law. This means that it's going to be it's going to be proportional to the concentration. This means the more of these photoexcited species that you have, the bigger the signal will be. Also will be related and will be proportional to the excitation coefficient. This is very important because you cannot directly compare the intensity of two different species because they can have like different excitation absorption. So you have to be careful with that. So this technique was invented by this person, Lord George Porter. He also, in the 50s, he also was a visiting professor in UCL in 1967. And in that same year, he got the Nobel Prize in chemistry. As a curiosity, if you go to Sheffield University, they have an EPRC facility where you can go there and you can measure some picosecond TAS. And they named this lab after Lord Porter. So what is TAS? TAS is a little bit complicated technique to understand sometimes. Some people struggle a little bit, but I find this way to approach it quite good. So before focusing on TAS, let's think on ground state absorbance, okay? Because almost everyone has measured ground state absorbance. In ground state absorbance, you have your sample, you shine with light of several wavelengths, and if you have organic material, in this case, you have your bandgap to discrete levels, and if the light matches with the energy of, with the bandgap of your material, you're going to absorb that light. Okay, for example, here the green, you can see like it matches with the energy gap, so you absorb that light. But if that light is going to be higher or smaller, the energy of this light is going to be higher or smaller, you are not going to absorb that light. So TAS is exactly the same thing. The only difference is that instead of having information about the ground state here, like in the ground state absorbance, what you're going to have like is information about the first excited state. How do we do that? With two different light sources. One is going to be the pump, and one is going to be the probe. The pump is going to be is going to be in charge of excite your system. So you're going to go from ground state to the first excited state. And then the probe is going to do a similar thing to ground state absorbance, but to your first excited state. This technique, as I said before, it has some time evolution. So if you establish some delay between the pump and the probe, you can see the evolution of your photoexcited state with time. So, but this technique, it doesn't measure the absolute absorbance of your photoexcited species. What it does is to measure the difference in optical density, delta OD, which is the absorbance after the pills minus before the pills. Okay, so what things can we measure with this technique? First, the photoinduced absorbance, which is the absorbance of your photoexcited species. Before the pills, you don't have anything of that species, so nothing can absorb. So before the pills, you have an absorbance which is 0. But after the pills, you have some population of these excited species. So that excited species is able to absorb sunlight. That's why after the pills, you have some absorbance. That's why delta D is positive. That's why here you can see how it's positive. This is the part that is going to be studied the most because the PIA of the different species is going to be different. So an exciton is going to have a different absorbance than a polaron, or can have a different absorbance because it's not always. So you're going to be able to probe the kinetics or the evolution with time of these different species by themselves. Okay, because for example, if we focus on the ground state bleach, and what is ground state bleach? Is when the probe wavelength is exactly the same as the ground state where your material absorbs. Okay, where your material has ground state absorbance. So before the POs, you have all your states in your ground state because you don't have any excitation. So you have absorbance. I mean, if you probe with the wavelength of the absorbance, you're going to absorb sunlight. Okay, so your absorbance before the PO is going to be positive. But after the pills, you're going to have less states in the ground state because, again, you're exciting your sample. You're going to have like some excited states. And after the pills, you're going to have less absorbance. Okay, so as after the pills, we're going to have less absorbance. That's why the delta D is negative for ground state bleach. Here, I want to remind some people, because in this positive and negative signal is because we are measuring delta D. In some papers, you will see it the other way around. Okay, the bleach, the ground state bleach is going to be positive, and the photoinduced absorbance is going to be negative. And that's because they usually use transmittance, and that's why it's different. But it's the most important thing here is that you understand why one of them is positive and why the other one is negative. The other thing is just a change of axis. We also can see some stimulated emission. Oh, sorry, I didn't say that in ground state bleach, you are able to follow the kinetics of the ground state. The problem with following the time evolution, the ground state, you can do it. The problem is like if you have several states, everything is going to recombine back to ground state. So you're not going to be able to distinguish between the different processes that happen to your ground state, which are going to be, like in some cases, a lot. Another thing that you can see is stimulated emission. Like for example here, the stimulated emission is just the PL of your molecule. Before the PLs, you're not exciting your molecule, so you're not going to have any PL. But after the PLs, you're going to have some excited, because you're going to be absorbing. You're going to have some excited states, and those excited states are able to recombine, and through this recombination, they can, well, not recombination, relaxation, sorry. Through this relaxation, they're going to be able to emit some light. This light is going to be recorded by the detector, and the detector doesn't know if it's PL or if it's absorbance, so it's going to detect more light. So for the detector, it will mean that this sample is going to absorb less after the pills. That's why delta D is negative. An easy way to check that you have stimulated emission is to check the shape of your region, of your feature, has the same features than the PL the PL of your material. Like here, for example, see, you have here 2 peaks, and here you have 2 different peaks. And the last thing that you can see in TAS is electroabsorption. The electroabsorption of the material, it's because we usually have materials, because we are working in OPV, of course, we have materials that can create some electric fields, because you are creating like an exciton or even porons. So these porons can create this charge. Let's say that this charge species can create an electric field. This electric field can create something called stack effect, and can change the energy levels of your material. Here you can see how the effect of the electric field of this species is going to decrease the energy levels of your material. Your HOMO and luminous are going to be changed. Well, actually, your ground state and your very excited state are going to be changed, are going to decrease. This means that the absorbance of this new species of the material after the electric field is going to be redshifted. If you do the delta D, which means the absorbance before minus after minus before, you're going to have like a positive signal of this negative, of this stack effect. Okay, so it's in the end, you're going to have like a red sheet version of your absorbance. So you have to be careful when you assign the different features that you see in your TA spectrum. Now I'm going to talk to you a little bit about the different setups that you can have for TAS. Mainly there are like 2, and they are different because they can measure different things in different timescales. The first one is the picosecond TAS. For the picosecond TAS, you will need a femtosecond laser, because of course you need a laser with a pulse width smaller than the actual thing that you want to measure. Otherwise, you were going to be able to only see the actual laser. You wouldn't be able to see like your different size species. And this is going to be your lower threshold for the measurement. In most of the cases, these femtosecond lasers have like 100 femtoseconds pulse width, so you're not going to be able to measure less than that. So this femtosecond laser is going to be, the beam is going to be split in two, because one of the two different bits which is split in two is going to be the pump, and the other one is going to be the probe. So the pump is going to go through the OPA, which is basically a photonic crystal. So you're going to be able to select the wavelength you want to pump your sample with, and you follow the green line and you hit your sample, and the probe is going to go through a delay stage. A delay stage is just a set of lenses that are going to change the distance that your beam is going to get through. Okay, because if you change this beam distance, you're going to change the difference between the distance that the pump and the probe can go. That's how in a picosecond TAS you are introducing the delay time. Okay, the bigger the distance that the probe does, the bigger the distance, the bigger of the delay time. Okay, and then this probe is going to go through a white light generator, because you need white light because most of these picosecond TAS are working with CCD detectors. These detectors you can set up one delay time with the delay stage, and it's going to record the spectrum at this delay time. Then the delay stage is going to change, and then you're going to record the spectrum at different delay time, and you're going to be able to have something like this, okay, with different delay times. And selecting one of the wavelengths later with your computer, you're going to be able to get the decay of this time evolution. This technique has an upper threshold, which is around 6 nanoseconds, 7, maybe 10, and it's due to the delay stage. Because if you want to go higher, if you would like to go to the microsecond, you will need a delay stage that can go, I don't know, like 200 of meters, and no one can afford like one of these super big laser tables of that size. It's impossible to do. It's quite unlikely to do. And this is more or less the setup for a microsecond TAS. Okay, in this case, you have two different light sources. One is going to be the laser, which is going to be the pump, and the other one is going to be the lamp, which is going to be the probe. Okay, both of these lights are going to hit the same part of the sample, and the probe light is going to get through a monochromator, which is going to be where we are going to select the probe wavelength. Okay, this light is going to hit a detector that is going to be connected to an oscilloscope. Because in this case, we are not going to use a CCD. This time we are going to use like a normal silicon array, okay, which is connected to the oscilloscope. So instead of having like the whole spectrum at one delay stage, what we are going to do is like selecting one probing wavelength, I'm going to have the whole decay for that wavelength. Okay, as you can see here. So the oscilloscope is going to control the delay time of your measurement, because you can tell the oscilloscope where is your laser, and your laser is going to be your time equals 0. Okay, and then the oscilloscope, because how oscilloscopes work, they know when it's 1 microsecond, 10 microsecond, and 100 microseconds. So it's how the decay is constructed. And then you select different, you do this experiment to several wavelengths, probing wavelengths, and then is how you get the spectrum of your excited species. The lower threshold of this one is given by two different things. First one is the laser, so the laser pulse has to be lower, but it has another issue, which is the oscilloscope. It's working because it's electrical equipment, and electrical equipment are not very good below the nanosecond timescales. That's why the threshold of microsecond TAS is usually from hundreds of nanoseconds, 100 if you are very lucky, to maybe, I don't know, like seconds timescales. So with two of them, you can have more or less almost every single time that you are interested in. Before I get a little bit deeper into the different processes that you can measure with this technique, I'm going to explain you a little bit about what process happen in a inorganic solar cell, because maybe you don't know anything about solar cells, or maybe you are you're been working with inorganic solar cell like perovskites or quantum dots. The main difference between inorganic and organic is that the organic materials have a very high Seldon-Binder energy because the electronical permittivity is like quite low. So the excitation-binding energy is like quite high, and that's why they are not able to absorb some light, create an exciton, and separate the charges. The organic materials need an interface, a donor acceptor interface, in order to split those charges. So in this case, a donor material, the blue one, is going to absorb some light. It's going to create a signal exciton, like you see here in the energy diagram. It has to diffuse until, to reach the interface. Of course, in the meanwhile, it can be relaxed to ground state, which is a loss process of solar cells. If you are lucky, this singlet is going to create what we call a CT state, which is a mixed state between the donor and acceptor state. And then this CT state is going to be able to split into free charges, and the whole is going to be in the donor, and the electron is going to be in the acceptor. Okay, of course, both CT and C charge states, also called parallels, can recombine by the CT state can recombine through Gemini recombination, which is monomolecular recombination, and the free charges can recombine through bimolecular or second order recombination. Also, another state that you have to think about in OPV is triplets. The triplets can be formed through intersection crossing of the singlet, and also the CT state can undergo intersection crossing to create a triplet CT state that can then go to be transformed into this triplet state. We have to notice that this triplet state is also a mechanism of loss pathway for OPV, so we have to have it in mind. So the timescales for these different species is in this slide, but this is it's something that's not written on stone, okay? So it's like some introduction thing. So not all signatures don't have these timescales, of course, but it's like some kind of orientation for you. The picosecond TAS can measure, like as we say, like hundreds of, or maybe 100 femtosecond to the tenths of nanoseconds. Meanwhile, the microsecond TAS can measure like hundreds of nanoseconds to the second timescale. So according to this graph, we can measure these processes, different processes in a solar cell. So the picosecond TAS can measure the things that you can see in your left, and the microsecond TAS you can measure the things that you can see on your right. The picosecond TAS can see that you can measure like way more things. The problem is like sometimes you can measure too many things, and it's quite complicated to understand. So sometimes, and also the good thing about microsecond TAS is like you can measure the actual charges. It's quite easy to measure charges, and also you can measure the charges, how they recombine and how they behave in your solar cell. And also you are using the timescales that are going to be useful for your solar cell, because the charge extraction on timescales of the devices are usually in the nanosecond, microsecond timescale. So both of them are very useful for their own things, and a mixture of both of them would be the perfect thing. So as I said before, the picosecond TAS sometimes is like super difficult. For example, you can see here in this spectrum, the evolution of the spectrum, you can see like you can have here one band, here you have another one, here you have another one, and if you see this one is like goes up and then goes down, the kinetics are not giving too much information because like too many things are going on at the same time. A decay then arise, this one stays stable. Sometimes what you do is like something that we call global analysis, GA. Next time you see a GA in a paper of spectroscopy, you can know that it's about this thing. And through this method, it's a mathematical method where you put some reference to it, and you end up with this. Okay, you put some reference. Our exciton is like this, and our boron is like this. So calculate me what else is going on in this material. Okay, and as you can see like how everything is more or less split, and the kinetics make sense. You have here a decay, a rise, a decay, a late rise, and everything start to make a lot more sense. The problem with this thing is, as I say, you have to put some references. So sometimes it's a little bit biased. Okay, so we have to be extremely careful when we do these things. So how do we do the analysis of the TAS? Okay, so the first thing we have to think about is most of the time different species have different peak positions. Okay, so the first thing is we have a new material is to check in literature if someone has measured it, and to check if you have exactly the same shape of your species. For example, the boron or the exciton. Also, you can have more or less an idea if you know what you think which species are you working on, because for example, if you can have some idea of the lifetime of those species. For example, the excitons, we know they're super fast recombining, and the charges are a little bit slower. Okay, for example, here in the spectrum we can see like this band decays super fast, and after 100 picoseconds there is almost nothing of it. But this one, for example, after 7 nanoseconds stays there. Okay, so we can do a fast assignment to, okay, so this band has to be the singlet because decays super fast, and this one decays much slower, so it has to be the charge. Of course, this one is a suggestion. Okay, so that's something that, okay, so you have done like a good 40%. Okay, so you have done a 40%, then you have to prove it, of course. What different things you can see also in TAS? The spectral shape of a species also can give you some information. For example, here Okita et al. In this paper, they found the P3HT. The P3HT is the polymer that everyone uses as a reference in OPB. Had two different peaks, one at 700, another one at 1000. And they saw that the peak at 700 decays faster than the one at 1000. So they suggested that this peak was due to borons that were free. Okay, they were able to recombine through pure bimolecular recombination because they were free, but these one were borons that were trapped. So their recombination was lower because they were trapped. Also, in this paper, the authors said that the shape can also give you an information about where these boron or these species are. For example, here they saw that in this material XND, they saw like how the borons can be in the bulk of the boron domain, and also if they are in the interfacial, they are in the interface between the XND and in this case of the P3HT, they have different shape with a band center of 1500 instead of 1400. Also, you don't really know if your spectral shape is going to be due to an anion, to a cation. So the best way to go on is to measure it with some references. For example, in this case, the authors measured this blend with a P3HT. The P3HT positive boron, it's very well known and it's absorbing at 1000. So they saw this band here, 1000, and they knew there was positive P3HT. So they saw a positive P3HT, so they thought, okay, so we have a positive species, we have to have a negative species. So as these species have the same kinetics that the positive species, so they saw that this is the negative boron of this XND material. Also, the selective excitation, because we can change our pump wavelength, it can give you also more information. Here, for example, again, in this paper, they use 700 nanometers as an excitation. Here, in this, at 700, P3HT doesn't absorb almost anything, and most of it is going to be absorbed by the XND. And still, we can see some P3HT positive boron. This means that even though we are exciting the other material, this material is going to be able to transfer the holes to the P3HT. So it can give you information about which processes are going on in your material, in your sample. Also, another experiment that you can do is the energy dependence, which is change the energy of the pump that you are using, okay, in terms of number of photons. That is very useful, for example, as in this case, that you can see like the number of photons that you were using, because the wavelength was exactly the same, was different in 3, 6, and 13, and the kinetics were exactly the same if they were normalized. Okay, for these 3 cases was very, very similar. This gives the information that you are having here a monomolecular process, because the kinetic doesn't change with the different, because if you have higher power, you have I said energy before, and it's power. It's very important. So if you have higher power, you will have more of your excited states. So you will have more of this concentration. So if it's a monomolecular process, the kinetics shouldn't change that much. The problem is, like as in real life, everything is a little more complicated than that. And in this case, if they increased too much the power, and they saw that this one was faster, because there was like some kind of singlet annihilation. This means that you have too many singlets in your excited state. So they can hit each other, and then they can relax through a process that was not present before, because there were not that many amount of singlets. And now I'm going to talk to you a little bit about the different species that you can see in TAS. For example, porons, because most of you are going to be working on solar cell, and maybe the most important species, the porons, because are the one that are going to be able to give you some energy to your device. Here you have the mixture of the joint of the femtosecond TAS and the microsecond TAS of one polaron. With the femtosecond TAS, what you are going to measure is the creation of these charge states, and also some of the annihilation through going back to the city state, and also some of the bimolecular recombination. But mostly it's going to be monomolecular. That's why it's more or less flat. It's going, sometimes in some cases it's rising, but some of them it's like quite flat, because you are creating more charges and recombining at the same time. By the microsecond, you can see only the recombination, which is going to be very useful. Because how porons behave in organic soil cell, they behave following a power law, like Jerry Nelson published in this paper. What is this power law associated to? It's associated to polarons trapped in a density in a tail of density of states. Okay, like here in this graph. This polaron is here trapped. So in order to recombine with this other charge, needs a little bit of extra thermal energy in order to go to the actual HOMO and be able to recombine. Okay, this will make this charge to recombine slower, but also the structure of this charge is going to be slower. So you need a compromise between these two. The alpha of this formula, which means also the slope when you plot the decay in log-log scale, is going to be between 0 and 1. The value of this alpha is going to give you the information about how deep these porons are trapped. The closer to 0, the deeper these porons are going to be, and the closer to 1, the shallower. And when this alpha is 1, it means that your polaron is completely free, it's not trapped at all, and that goes through pure bimolecular recombination. Also, I'm going to talk to you a little bit about triplets, because I've seen some papers where, as the solar cell is efficient, they don't address anything about triplets, and they do have triplets. So we have to be careful. The triplets also recombine through a monomolecular process, so they are going to follow a mono-exponential decay, like the singlets, and they are going to be linear if you plot them in the log linear scale. Another interesting thing about triplets that in a microsecond TAS, you are going to be able to do the oxygen quenching experiment. What is the experiment? Because the triplet can react with the oxygen atmosphere in order to quench these triplets. As you can see here in this graph, you can see how the nitrogen, the signal is beautiful, big signal, beautiful decay. When you add some oxygen to your sample, the signal gets totally quenched. But when you take out all the oxygen again and put some nitrogen, it goes back and you still have more or less the same signal in both in size and kinetics. This is very important that you do this recovery part, because that's going to give you the proof that this process is reversible, okay? Because the oxygen quenching is a reversible process. Otherwise, you don't know if this quenching is due to oxygen quenching or maybe because the oxygen is destroying your sample, okay? Also, you have to be careful when you do this experiment, because this experiment only works if the oxygen level, which is 0.98 EV, it's below your triple level. In some cases, like very low backup material, the triple level is quite low. So this triple level is not going to react with your oxygen. So that's why in this case, the authors showed that there was no quenching at all of the triplets. And now I'm going to show you a little bit about CT states, because CT states are a little bit interesting because they're not that much studied. And here are the reasons why. The first one is because they usually have a low extinction coefficient. It's quite difficult to see one because the absorbance is going to be very low. And also, even when you see them, the absorbance is going to be very similar to the ones of the porons and or anions and cations. This is because the CT state, remember that it was a mixed state between the acceptor and donor. So sometimes have a lot of character of either one of those. For example, here, the authors saw this in this paper, this feature, and they assigned this to PC60 anion. But then doing more tests, they realized that this was not due to the PC60 anion, because was not due to the PC60 anion, and they found out that it was a CT state. Okay, and exactly with the same absorption spectrum than the actual PC60 anion. So the conclusion, I show you more or less the basic of transient absorption spectroscopy. I explained you a little bit about how to set up, or how is a setup of picosecond TAS and microsecond TAS. Also, I give you some insight about the analysis of the different species that you can see in OPV, and a little reminder that this technique is not only useful for OPV or for solar cells. You can also use it for LEDs, for LEDs, for water splitting, for photocatalysis, photosynthesis, etc.